This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1150, recorded on September 19th, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York, Daniel Griffin.
Hello, everyone.
Looking very scholarly with the books and the bow tie. Daniel, what's on it?
So I have Ebola again. So
it's not Friday. Yes, it's not going to be a clap.
Not Friday.
Okay.
Actually, I've got some things going on tomorrow. I'm actually going to wear a coronavirus, beta coronavirus bow tie tomorrow on a Friday. Imagine that.
Imagine that. Well, it is spreading, you know.
Yeah. So. All right, well, let's jump in. We got a lot to cover. We're recording, I guess, late here on a Thursday night. And I will jump in with my quotation. They conceive a certain theory, and everything has to fit into that theory. If one little fact will not fit in, they throw it aside. But it is always the facts that will not fit in that are significant.
Daniel, do you think that's how scientists sometimes work?
The way this is written, it's a little ambiguous. What is the it that they're throwing aside? Are they throwing away that little fact that doesn't really fit? Are they throwing away the theory? Well, as long as it's the theory, I'm good with it. But there certainly are scientists and clinicians where it's the facts that get thrown away, the actual facts. Yeah.
It's interesting. Yeah. What is she talking about? The thing that doesn't fit or the theory? Yeah.
Yeah, I mean, I have to say that the reading I like is it's the fact that gets thrown away. So,
well, how do you know when to throw it away, though?
You never throw the fact away, as we learned from Agatha Christie. It's the facts that don't fit that are often the most.
You keep them on the side and ponder them.
Yeah, why does that not fit? What is wrong with our theory?
That's right.
Why did we not anticipate?
You just said it. What's wrong with our theory? That's the key.
Yeah, make
questioning.
Sure question your hypothesis, your idea, your premise. All right, well, we've got West Nile virus. I wanted to mention this because we just had a couple deaths from West Nile virus in New Jersey. So just to keep some awareness on this, as we see deadly mosquito-borne infections. Now, what we read in this, it's actually a CBS News piece, is we have so far had 8 total cases in New Jersey, 7 were hospitalized, experienced swelling of the brain or swelling of the lining surrounding the brain and spinal cord. But I have to say, I suspect this number of cases and severity distribution is probably due to a lack of diagnosis of milder cases. I mean, just a lack of diagnosis of cases in general. Because, I mean, just at the one hospital where I spend a lot of time, we've seen, 3 cases just in the last few weeks. So it's really a question of people don't often look for things. And then you only see, the worst. Polio, it sounds as though things are progressing well in Gaza with respect to the polio vaccine. We had a report this past Monday that the polio vaccinations had already reached 90% of goal. But on a not so good note, at the same time, we heard that the Taliban has suspended polio vaccination campaigns in Afghanistan. Now, I'm trying to get a little more understanding, and maybe Vincent, you have some ideas, but my understanding there's been discussions about moving away from this house-to-house vaccination approach and instead having immunizations in places like mosques. So I'm hoping maybe this is just a shift in the approach rather than an end to the vaccinations.
Yeah, I don't think the Taliban likes the house-to-house approach and wants to shift it to mosques. That's my interpretation, but they're still stopping for the moment. They're suspending, and I read a quote somewhere that they think it's an American plot to sterilize their children, so they're not very happy about it. So this is all misunderstanding again.
Yeah. I mean, it's really important to keep politics and everything out of vaccination campaigns. But no, that's been an issue. That was an issue in the smallpox campaign it was actually Vietnam, and the French were involved. There's a lot of mistrust. So there was a lot of issues there. There's a lot of parts of the world where you want to really be sensitive to what the local people are concerned about, and really the importance of partnerships, letting them be involved and actually modify the campaigns to what they're most comfortable doing. So I will try to be optimistic. Dengue. LA County Public Health just announced 2 more locally acquired dengue cases. So this is LA. So now California, Florida. So in this, we have a cluster. So this raises the number in that cluster to 3. The 2 latest patients are from Baldwin Park, the same area where a first case was reported last week. We read that the Aedes mosquitoes are common in LA, but local dengue cases or diagnosed local dengue cases are rare. But back in 2023, 2 similar cases were reported, one in Long Beach, the other in Pasadena.
So this is interesting because locally acquired means there's no travel involved. So there's some kind of reservoir of the virus there.
Yeah, I mean, that's a concern. And we had a number of years ago, a teenage boy, I think it was in Huntington, just a few miles from where I live, no travel, nothing, gets dengue. He's quite the astute clinician to actually even think to order the proper testing and pick that up. So.
Yeah, when it's so rare, it's difficult, right?
Yeah. Why? How did that get into your differential? You'd say never traveled. So that's always our challenge.
But you would say, Daniel, that now that we have local dengue, say in LA and other places, it should be on the differential, right?
Yeah, and hopefully, a lot of times I think people say, "Oh, medical school, we're learning all this stuff, and I'll never see it." Well, you might not diagnose this if you didn't realize. And there is a rather characteristic rash that we often see in dengue, and so that might be a little bit of a trigger. The timing of the fever and the fever abates, and then a characteristic rash. You may clinically raise your suspicion, even though, it isn't necessarily endemic otherwise. Flu, a reminder, sort of a morbid reminder, but a reminder to schedule that influenza shot for you and your loved ones, including your children. It's about this time every year we sort of hear what were the total deaths from last year. And we're hearing from the CDC that last year, 199 children died from flu. So we're back. That was the level that we were at in 2019, 2020. Among the fatal cases, 73 were younger than 5 years old. 126 were aged 5 to 17. So some 18-year-olds died. Interesting that about half were influenza A, right? Half were influenza B. They do mention that 3 children had co-infections, influenza A and influenza B. Now, 2, I think, really important points. Of the 189 children with health status information, the majority, just over the majority, 51% did not have a single underlying medical condition. These were, up until this point in time, otherwise healthy children. Now, of the 158 children that were eligible for flu vaccine, for whom the vaccination status was known, 83% were not fully vaccinated. So really suggesting these are mostly preventable deaths. So I know a lot of people, concerns, why am I vaccinating my children? Well, 200 children, died last year from flu. All right, mpox. We heard on September 13th that the WHO pre-qualifies the first vaccine against mpox. WHO announced the MVA-BN. So this is the modified Vaccinia and CAR vaccine made by Bavarian Nordic, and it's added to the pre-qualification list. So a little bit of terminology. What is a pre-qualification, emergency use? These are mechanisms used to evaluate quality, safety, and efficacy of medical products, such as vaccines, diagnostics, medicines, and product suitability for use in low, middle income country contexts. So these PQ, pre-qualification, and EUL, emergency use listing, these assist decision for international, regional, and country procurement by UN and partner procurement agencies. So little context here, the available data, we've covered this. It shows about this 80%. So what we're seeing here is available data shows that the single dose given before exposure has an estimated 76% effectiveness in protecting people against symptomatic mpox, with the 2-dose achieving an estimated 82% effectiveness. And we talked about the studies sort of really in the same ballpark, but also the fact that even if you end up getting symptomatic mpox severity. So great impact on severity. Like folks that get vaccinated don't usually end up requiring hospitalization. Good safety profile, vaccine performance consistently demonstrated. Also seeing, real world evidence here. So the WHO, even a little flexibility. There might be situations where a single dose use is applied in supply constrained outbreak situations. And still exciting news in this area. We did hear on September 18th, that's yesterday, the day before we record this, and a few days before you hear it, that Gavi, the Vaccine Alliance, and Bavarian Nordic announced an advanced purchase agreement to secure 500,000 doses of the Jynneos mpox vaccine for African countries. And they're going to be delivered this year. All right, still in the mpox area, we got the publication TECO Verat Use Under Expanded Access to Treat Mpox in the United States, 2022 to 2023, published in NEJM Evidence, New England Journal of Medicine Evidence. So this is really kind of a descriptive publication. So TECO Verat, this is this antiviral for mpox, was prescribed to over 7,100 patients in the U.S., really most often for lesions in sensitive anatomical areas, such as anogenital lesions. Now, the demographic clinical characteristics mirrored those of patients worldwide. So you sort of get a nice ability to look across, even though we're only looking at this particular population. Now, among the 7,181 patients with returned intake forms, 1,626 also had these outcome forms, right? So we're sort of getting to smaller numbers, but many patients with severe immunocompromise, so HIV, CD4 counts less than 200, received multiple courses of TECO Verat. Now, overall, 223 serious adverse events and 40 deaths were reported. Now, most of the serious adverse events were among patients who were severely immunocompromised. One have experienced hallucinations after TECO Verat, being given at twice the standard dose. So I think just the takeaway here I want to say is we're learning about TECO Verat. We're still waiting for some really good, solid human efficacy data.
So, Daniel, we should know how late after the rash appears that you could give it and have it be effective, right?
I think that's important. What's a challenge with mpox is that it seems that as soon as you get into the prodrome, right, so you start to have some symptoms, don't necessarily have obvious lesions, there's already, case, transmission, documented transmission. You also, even when it's the vesicles are gone, you've got scabs, you do not yet have full intact skin, we're seeing spread to others. So sort of evidence at both extremes here that there's still replication competent virus here. So it is sort of interesting, like when do you jump in? And we really need the data on when does it make the most difference, right? Because we would really anticipate that the sooner you start, the better outcomes we're going to see.
But many of these patients are immunocompromised, so that also plays a big role. It's probably going to be less effective in that cohort, right?
I would agree. And it may actually be that different durations, right? So
sure.
All right. Bird flu. Oh my gosh. We're still talking about bird flu. We read from the CDC, one new human infection with a novel influenza A virus was reported by the Missouri Department of Health and Senior Services. The patient was infected with an influenza A H5 virus. So we read that the patient is older than 18 years, has multiple underlying medical conditions. Patient developed symptoms during the week ending August 24, 2024, hospitalized, since recovered. A respiratory specimen collected from the patient tested positive for influenza A at the hospital. They forwarded the specimen to the Missouri State Public Health Laboratory. And the CDC confirmed the infection was caused by an influenza A H5 virus. Now, a subsequent investigation by state, local public health officials did not find any direct or indirect contact with wild birds, domestic poultry, cattle, including no consumption of raw dairy products or other wildlife prior to the patient's illness, right? So that has everyone, what's going on here. Now, the other got people sort of hackled up was one close contact with the patient was also ill at the same time. And initially we read was not tested and has since recovered. we read a little bit more, right? So we read in the New York Times article that someone who lived with a Missouri resident infected with bird flu also became ill on the same day. Coincident or causation? So, we go on to read, it's getting all exciting. And, maybe the Agatha Christie was a good quote up front. The disclosure raises the possibility that the virus H5N1 spread from one person to another, experts said, in what would be the first known instance in the United States. Now, on Friday night, this is last week, CDC officials said that there was no epidemiological evidence at this time to support person-to-person transmission of H5N1. The coincidental timing of the illness, especially outside flu season, concerned independent experts. But then we read this article by Brenda Goodman at CNN that a second close contact at healthcare workers developed mild symptoms but tested negative for flu.
This is the problem with he said, she said. The CDC that says there's no evidence. What does Barbara Goodman know?
Well, so Brenda Goodman's actually the one who points out that the test was negative. It's actually our buddies at the New York Times that are speculating about human-to-human transmission.
So the thing is, outside of the U.S., there has been limited human-to-human transmission, especially when people live together, right? So this would not, if one person had it and the other was living with them, it wouldn't be surprising. But we have so little information that it shouldn't even make the news.
Yeah, but we can speculate. It's exciting.
Of course. So according to Agatha Christie, what do we do with this piece of information?
Exactly. We need more. So, all right. So let's jump into COVID here. We always bring up our map, what's going on. And this is deaths, the percentage of provisional deaths due to COVID-19 in the past week, looking at the different states in the U.S. And now not only Kentucky, but we also have West Virginia at that 4 to 6% of all deaths due to COVID. Lots of 2 to 4% in our country as well. Now, a couple interesting things, Vincent, you and I have been following this is the wastewater. We're going to look at this a couple ways. So one is our traditional way, and it looks like it in most areas, we might be on the way down and kind of, out west, it was down and then looked like it was rising. In the south, it looks like it's on the way down. Nationwide, maybe a little down and then rising. In the Midwest, just kind of plateauing. Now, I kind of went back and I looked at it really data all the way back to January 22 to kind of get this pattern of what we're seeing. So for instance, last year, right, we saw a little bit of a summer bump and then we saw a winter peak. Now we're seeing actually this peak here, sort of end of summer into fall, wastewater that was as high as last year. So we'll see what happens. The bets are in.
So, it's funny, in 22, we had a summer peak.
Yep.
Right. And then we had another one in the winter. Yeah. Then in 23, a small one at the end of the summer, one in the winter. And now we have a big one in the summer. So there's always been a little summer activity.
Right? Will we see,
Yeah. So there's a little summer activity every year, but it varies in intensity, it looks like.
Yeah. So. And actually, I'm going to put in a new link for wastewater this week, so people can take a look at this, and it's really nice, actually. So this is data where you can actually you can click on the left, and you can look at SARS-CoV-2, you can look at RSV, influenza, human metapneumovirus, norovirus, mpox, rotavirus, EBD68. Maybe we'll talk about that. Candida auris and Hep A, and they actually have with the SARS-CoV-2, you've got this map where you've got medium out west and high, really basically most of the country. Then you go to EBD68 and actually medium across the country. I don't know if you had any comments on that, Vincent.
So EBD68 is one of these late summer, fall infections. We haven't seen a lot of cases in the past few years, although now they've been picking up. But the key here is that enterovirus D68, a member of the same genus as poliovirus, has been associated with acute flaccid myelitis. But in this current activity scenario, there have been no reported cases of AFM which associated with EBD68, which is good.
All right, we'll keep our eye on that. All right, moving to vaccines. And remind me, Vincent, if I forget, because people always ask, when should I get my vaccination? So we have the article, Effectiveness of Updated 2023-2024 Monovalent XBB.1.5 COVID-19 Vaccination Against SARS-CoV-2 Omicron XBB and BA2.86JN1 Lineage Hospitalization and a Comparison of Clinical Severity, IV Network, 26 Hospitals, 18 October 2023, 9 March 2024, published in CID. And we spent a time trying to sort of sort out what information we actually have here. So here these investigators analyzed patients hospitalized with COVID-19-like illness at 26 hospitals in 20 U.S. states, admitted 18th October 2023 through 9 March 2024, sort of thing. We're thinking like last winter season, roughly, right? They're going to use a test-negative case-control design, and they're going to estimate the effectiveness of an updated 2023-2024 monovalent XBB COVID-19 vaccine dose against sequence-confirmed XBB and JN1 lineage hospitalization, right? So we're going to get to look at an XBB vaccine and how did it do with the two different lineages, right? Because we talk over time, is it about time that goes by? Is it the lineage? Is it the virus that really makes the difference? So now we only, I introduced the word, we only have 585 case patients with XBB lineages. We only have 397 case patients with JN lineages. So I would love to have more, but okay. We've got 4,580 control patients. Now, Vince and I will go through this figure here in a minute, but the vaccine efficacy in the first, let's say 90 days, 7 to 89 days after receipt of an updated dose, they're going to say 54% against XBB lineage hospitalization and 32.7% against JN lineage hospitalization. Odds of ICU admission or invasive mechanical ventilation and death were not significantly different among the JN compared with the XBB lineage. So in the discussion, the authors point out that these findings indicate that despite substantial genomic divergence of JN lineages from XBB, updated COVID-19 vaccines continue to provide protection against COVID-19 associated hospitalization. Wide confidence intervals for some estimates precluded confident assessment of the extent to which vaccine efficacy differs by lineage.
Daniel, what is the age and comorbidities of these patients? Do we know?
It actually is in the
Are they matched, in other words?
Yeah. Yep. So they are matched. That's at least good.
Okay. Yeah, I mean, I suspect that it doesn't matter which booster you've got. I think you're going to have those T cells protecting you, unless you're really, really ill and are quite old, because then you won't have memory T cells.
Yeah. I mean, if anything, it's encouraging. It really looked like, even when it was, the JN lineage, the JN1 lineage versus the XBB lineage, it looks like we're getting some, overlapping sort of efficacy here.
I want to find all the people that got just the original vaccine, 3 doses, and see how they've fared over the years if they have stayed out of the hospital.
Yeah.
I bet they've done fine as long as they're healthy.
Well, it is interesting, and I think this was actually something I talked about when I was in Taiwan. If you look at individuals under the age of 65, was actually the study I was looking at in Taiwan. There's this phenomenon where you get your first 3 vaccines. If you get reinfected, you tend to do fine. Your chance of ending up in the hospital goes down. Sort of this concept of boosting. Now, the whole interesting issue was if you then looked at people over 65 with medical issues, it was kind of the opposite. You actually saw that things were sort of chipping away with each infection. So yeah, it is important, as you bring up. You got to know, like, who are we talking about? What age? Comorbidities? So really, the importance, I think, of that, personalized discussion, with your medical provider about what you need to do and what your risks are.
In fact, in this paper, it would behoove them to stratify the results by age groups and see if you get a higher protection or VE under 65 versus over 65. That would be quite compelling that the T cells are involved, and other mechanisms maybe also.
Yeah. And I bemoan, I say, 585, 397. I'd love to have more. I mean, you need larger numbers to really narrow those confidence intervals and really.
Well, this is where in countries that have, what is it, electronic health records.
Nationalized
then you could just access them and look at it.
Yeah, somewhere like Norway, where whether you like it or not. All right. We also have the article, Early Robust Mucosal Secretory IgA, but not IgG, response to SARS-CoV-2 spike in oral fluid is associated with a faster viral clearance and COVID-19 symptom resolution, published in JID. So here they're going to investigate the role of oral mucosal antibody responses in viral clearance and COVID-19 symptom duration. So just a quick reminder for our listeners, what are we talking about? So IgA is the main mucosal antibody type, as opposed to IgG, that's the main serum type. So think particularly of the secretory IgA as being this antibody that we're hoping to have at our mucosal surfaces, such as the whole upper and lower respiratory, our GI tract. Now, the way they did the study was to give participants with PCR-confirmed SARS-CoV-2 infection this oral fluid testing. So you're going to get the nasal swabs, you're going to get all these follow-ups for the symptoms. They found that high and moderate oral fluid anti-spike, secretory IgA post-infection was associated with a significantly faster viral clearance, so dropping an RNA copy number to be, and symptom resolution across age groups with effect sizes equivalent to having COVID-19 vaccine immunity at the time of infection. So those with the, sort of break it down, but those with high and moderate anti-S secretory IgA cleared the virus 14 days, and they give us this confidence interval, and recovered 9 to 10 days earlier than the folks that had those low IgA levels. So delayed and higher anti-S IgG was actually associated with significantly longer time to clearance and recovery. They're sort of interesting.
Yeah, it's not surprising that an IgA would be doing this. And we know that everybody is slightly different when they respond. So you're going to have high responders and low responders. So it makes sense.
Yeah, I think maybe in the future, we're going to maybe be asking for more information from our vaccines, right? Like we know about like serum neutralizing antibodies. We know about Th1 versus Th2. Are we going to start requesting like, hey, can you give us, mucosal immunoglobulin?
But Daniel, fundamentally, why would secretory IgA be better than secretory IgG? Because IgG does make it into the mucosa. Is there something better about IgA?
It's interesting. There are people out there, it's like 1 in 700, 1 in 1,000, depending upon your sort of genetic background, that actually do not make IgA, right? So they actually compensate with the other antibodies. So in most people, that's going to be 99.99% of the time, it's IgA that we're measuring when we're looking at mucosal surfaces. But no, if you're one of those folks that are using, a different immunoglobulin at your mucosal surfaces, I don't think the actual, antibody would matter.
It's the timing.
Yeah, it's probably timing. All right. Remember passive vaccination, right? PEMGARDA for that 70% risk reduction of developing symptomatic COVID-19 compared to placebo based upon those early trials. COVID early viral phase, the treatment guidelines have not changed. NIH, IDSA, number 1, Paxlovid. And there's been some nice commentaries out there about like, be careful when you look at these meta-analysis and they start including preprints and stuff. We now have 100 plus articles, randomized control trials. We have real world efficacy data. Paxlovid, particularly if you can get it the first day, within the first 3 days, really up to the first 5 days, reduced risk of progression to hospitalization and death, really compelling data in the high-risk population, growing amount of data in long COVID as well, which we'll continue to share. Number 2, remdesivir, 3, molnupiravir, 4, convalescent plasma. And remember, when you're sick, you are contagious. 3, so they're moving on to the early inflammatory week. Remember, this is that bad week, right? My daughter's off at college, Eloise, and with the they now call it Pledge Week or Recruitment Week. I don't know, whatever the sororities and Greek organizations do. They've renamed it from. We used to call it, I think, Pledge Week or some other
Rush.
Rush. That's what it was. Yes. Yeah, for those who don't know, I was in a fraternity at University of Miami. It was really more of a scholarly gathering.
Which one, Daniel?
I was an Alpha Tau Omega.
Okay, good.
So, but it's so interesting. I'm getting descriptions of folks that are, can you believe this? They're having Paxlovid rebound, but they never got Paxlovid.
Gee, they just had COVID, right?
Imagine that. They got COVID, they started to feel better, and oh my gosh, that second week, they felt horrible. Anyway, so yeah, just to remind people, it's that second week when people can really feel rotten. And that's when we often see the hypoxemia, hospitalization, and death. So, in general, you weather the storm, but for folks, right patients, right time, there are roles for steroids, anticoagulation, pulmonary support, remdesivir still in the first 10 days, immune modulation. And interesting, a little bit more tocilizumab being used in Taiwan than we probably use here in the States. And all right, this is an interesting one. So we'll spend just a little bit of time. And, I suspect we'll get some letters about this, emails. But the article, Prevalent Metformin Use in Adults with Diabetes and the Incidence of Long COVID and EHR-Based Cohort Study from the Recover Program, published in Diabetes Care. So these are results of a retrospective cohort analysis using the National COVID Cohort Collaborative, N3C is important. We're going to talk about N3C. That's the first cohort. We also have the Patient-Centered Clinical Research Network. That's the PCORNet, electronic health record databases. And they're looking at individuals with type 2 diabetes who were or who were not taking metformin when they were diagnosed with COVID-19, right? So not that complicated ramp up and one and this and that. It's just, hey, you got COVID-19, we're on metformin or not. We got the two databases. So kind of mixed depending on which database they look at. So just looking at the NC3 data, we get a suggested 15 to 20% decrease in PASC, being on metformin. Looking at the PCORNet database, it might be decreased by 13%, it might be increased by 4%, but we see sort of this overlapping confidence intervals. Looking at raw percentages, we might be talking about a 1% overall reduction. So the raw differences are not quite as impressive as what we're seeing here.
So based on this, Daniel, would you recommend metformin for PASC?
So what they're really looking at here is if you've got diabetes, does metformin reduce your risk of getting PASC? So not really a treat. I think that's important. It's not recommending this as a treatment for PASC. It still also is unclear that if you get acute COVID, should we jump in with that complicated metformin, or do you just do Paxlovid, right? Probably has pretty similar data. And is metformin something we should be using with Paxlovid? Something we should be using instead of Paxlovid? I would say no. I think the one of the authors on our COVID Out trial was sort of thinking of metformin as like the poor man's Paxlovid. Maybe there's some antiviral, maybe there's some effect here, but the data we're getting with Paxlovid are much more impressive.
So here, if you are diabetic and you're on metformin, the protection against PASC is minimal.
It's minimal, but there may be some. Maybe some. All right. And no one is safe until everyone is safe. I've been saying that now for a while. It's been true. It continues to be true. So I want everyone to pause the recording right here. Go to parasiteswithoutborders.com and click on that donate button. We're in the middle of our Floating Doctors fundraiser, really right in the middle, actually, August, September, and October. We're going to double your donations. Hopefully, we're going to get up to a potential maximum donation of $20,000 to support the great work that the Floating Doctors are doing.
It's time for your questions for Daniel. You can send them to daniel@microbe.tv. Eli writes, are masks recommended for commercial flights, or does the turnover of cabin air once in flight mean they are not needed?
Ellie or Eli, not sure how you pronounce that, but, we've actually, over the last few years, we've shared a number of studies where actually the duration of that flight, and it's sort of a binary. Once you get above 5 or 6 hours, you really start to see an increase in the number of folks that end up getting COVID-19, getting infected with SARS-CoV-2 on those planes. So it isn't just the getting off and on the plane. It's unfortunately the folks around you. We are seeing transmission. So you gotta ask the question if you're on one of these flights and people around you, and particularly there's a lot of folks with COVID. There's a lot of SARS-CoV-2 on the planes, as Noah Khan tells us. Then you might consider wearing a mask.
I mean, they could be sitting next to you shedding and they have no symptoms. Who
They don't
knows?
even know. Yeah, they don't.
And then the guy in front starts hacking away, and the HEPA filtering, and got to stop that.
Yeah.
And then they walk down the aisle and cough as they reach you, right?
Oh yes.
So nothing is safe. No one is safe until everyone is safe.
Yes.
Anita writes, while listening to the most recent TWiV, you responded to an email about hepatitis B vaccination. I thought it may be worthwhile to mention Heplisav B as an option for your listeners. What's that? Is that a kind of Hep B vaccine, Daniel?
So it's I'm going to say relatively new 2-dose option, right? So historically, all our Hep B vaccines have been 1, 2, 3. Heplisav is a 2-dose. So I'm really kind of curious to see, how this gets introduced. But yeah, it's great that you can go ahead and get that full vaccination. It's an adjuvanted vaccine.
Linda writes, I'm a 50-something who will be a poll worker on Election Day. I would like to get a COVID booster prior to this. What would be the best timing? Let's take that first.
Yes. Okay. So that's great. Thank you for reminding me that we should be talking about timing of vaccinations. So as we've talked several times, 2 weeks prior is when you're going to hit that, that peak. So if you really want to have peak on Election Day, you want to get that booster about 2 weeks before. So a lot of people are sort of looking at, when am I going to get my booster here this fall? You want to be thinking, and it's going to be a little bit individual, right? Okay, I had COVID recently. I got to wait 3 months, and then you do it. Hey, I'm really going to be laying low, but I'm going to be really spending a lot of time with, family and friends, and in November, December. Okay. So you might want to think about early November and try not to get it in the meantime. Maybe the high holidays are coming up for you. So boy, you probably want to jump in there and get it now, or be careful with the high holidays and then be thinking about these other times. We really think these are, they call them new vaccines, but they seem to be boosters. They seem to be giving us a period of, we'll say, 4 to 6 months of increased protection. Even the study that we talked about earlier, when you look at past that 90 days, you start to actually see protection drop, even with regard to outcomes like hospitalization. Flu, right? We'll talk about that in the same breath. Flu, we have data now for, a number of years where, you get that peak about 2 weeks afterwards, and you're losing about 10% of that protection against medically attended flu per month. So think about kind of the timing. The interesting thing about flu, it's sort of a word of caution, an update. It used to be pretty reliable, right? That flu would come like December or maybe come a little bit later, but we're seeing earlier flu. So it used to be, maybe September's a little too soon, and that was true 6 years ago. But now you want to start thinking about keeping track about what's going on with flu.
All right. And Linda's second question is, I've never had chickenpox. I've had 2 chickenpox vaccines. The second was about 20 years ago. I had a negative varicella zoster IgG titer, test in 2021. Do I still need to get a shingles vaccine?
So this is a great question, right? So the reason I say it's a great question is that what we do now is the chickenpox vaccines that we're using, these are actually replication competent attenuated vaccines, what some people would say were live vaccines. So there are described, it tends to be pretty rare, that people will get shingles from those. So, in general, we're starting to get to where we really have to address this question. In general, very, very low risk. So boy, even if you reduce that risk by 90%, you've already started with a very low risk. There's no harm to getting the shingles vaccine, but you're really, as far as efficacy, a 90% reduction in what's already a very low number. Now, at some point, I would love if we moved away from using a replication competent virus, right? Like let's stop putting this virus out there, and let's just actually go ahead and use our protein-based shingles vaccine.
Shingrix, right?
Shingrix, yes.
For everybody, for kids and adults.
Yeah,
right?
Just use it across the board. But again, we've got to do the science. We've got to see.
Jay writes, I'm disappointed by Emily Schmall's piece in the September 3rd New York Times, The Best Time to Get a Flu Shot, in which she writes, Experts said that for most people, getting a flu shot at the start of September may be too early to provide protection that will last throughout the flu season, end quote. What's disappointing is she did not provide any evidence to support this. There was expert opinion without naming the experts. I've not found any evidence that getting the flu vaccine later in the season, say October or November, leads to better, meaningful outcomes. If I'm missing something, I do want to know. Do you know any evidence that getting the vaccine later leads to less disease or death?
Yeah, so Dr. Gladstein, we just actually mentioned this a little bit earlier. So there's a number of studies where we're sort of getting this 10 to 15% per month decay in the protection against medically attended flu. Now, that is interesting. What about what we really care about is people ending up in the hospital, people dying. I think we've shared in the past that 90% of people that succumb to the flu were not vaccinated. And we even shared like the data on children, like 85% of the children that died last year, 83% I think was the actual number, did not get that vaccine. So the flu deaths that we see are largely the unvaccinated, not necessarily that, oh, they got it too early in the season. So yeah, I agree with you. When experts are speaking about something, it's really nice when there can be a link to the actual studies, or even at least knowing who those experts are.
Scott writes, hello, Dr. Daniel. Scott here. Maybe it's Scotty.
Is this from Trek? You think that's...
I'm going through my second infection in 2 years. I've taken all the boosters whenever available. And you'll see this is COVID because he says, the symptoms this time were incredibly mild. I still got Paxlovid, which was discouraged by the urgent care medical staff I spoke with, bringing up concerns of rebound. Being a regular consumer of TWiV, I pushed back and got the prescription. This has got to be the scrappy Scotty, you know?
Yes.
The last time I took Paxlovid, I did experience the situation where I tested negative, and then several days later, I got some sniffles and retested and was positive. In preparation for this possibility, my question, am I contagious if this happens again? I will be at day 8 tomorrow, and I'm symptom-free and testing negative.
Scott, if you listen, what are you doing retesting? We've covered this. Look at your calendar. Stop sticking things up your nose. People can get, positive antigen tests. They can get positive nucleic acid amplification tests after those, most contagious first 5 days. You're asking for trouble by sticking stuff up your nose. Most of the contagion, 85%, happens in those first 5 days. We really don't see documented transmission after day 8.
All right, now we received a lot of emails because apparently the Florida Department of Health sent out their latest COVID vaccine guidance, in which they do not recommend mRNA vaccines. And one of them here is from Laura. No doubt I am not the first person to send you this link to the Florida Surgeon General's updated guidance for COVID-19 boosters for fall and winter 2024-25. As someone who taught and researched scientific and technical communication my entire career, not to mention courses in research methods, I find the rhetorical approach of this new, quote, guidance, unquote, to be, shall we say, interesting. Links to external studies from credible sources, such as the Cleveland Clinic, provide readers with a sense of credibility. The logical flow then creates an overall narrative that invites trust. Yet logical fallacies, incorrect interpretations, and much cherry-picking abound. I recognize there are too many issues to be addressed on TWiV, but I wondered if you could perhaps speak to two of the most egregious. One, DNA integration, and what the memo suggests is the risk that DNA integrated into sperm or egg gametes could be passed on to offspring of mRNA COVID-19 vaccine recipients. And two, the claim made about the Cleveland Clinic study that as efficacy waned, studies showed that COVID-19 vaccinated individuals developed an increased risk for infection. I find memos such as this, which do not on their face appear to be misinformation, to be more worrisome than communication that is overtly not creditable. In order to avoid legal issues, many physicians in Florida don't know what they can safely say to their patients, except that when it comes to COVID boosters, you are on your own. To quote you, what does one get when you mix science with politics? Politics.
All right. Thanks, Laura, for bringing this to our attention. This is, there's a bit of a history with this. And it's a challenge, right? So we'll deal with the science. So this whole, concern or raised concern of DNA integration, that was actually, when you go back a little more than a decade, when people were considering different vaccine technologies, different nucleic acid vaccine technologies, the DNA-based vaccine technologies, this was raised as a concern. If you've got DNA and you got to get that DNA into the nucleus, could that DNA then integrate? Now, mRNA, right? And this is basic science, but important basic science. It's about where that mRNA needs to traffic, where it needs to go. The mRNA just needs to get into the cell, into the cytoplasm. You're done. You're all good. That's where the mRNA serves as a template for the protein production. In this case, the spike protein production. So this was actually a big driver towards mRNA COVID-19 vaccine. So really, the exact opposite. Now, the other claim they make about COVID-19 vaccinated individuals, I remember this study. And again, it's interesting because I spent a little time looking through. There's sort of like, have your cake and eat it too, where they point out that the point of the COVID-19 vaccinations was to prevent disease. And when they first introduced, Omicron was circulating. So they tried to say, oh, well, they prevented disease, but only they were talking about Omicron back then. But here we're talking about risk of infection. But as we've talked several times, that's not what vaccines are about. Vaccines prevent you from getting disease, from getting severely ill, from ending up in the hospital, ending up in an ICU, ending up not surviving. And the data is very clear. I guess, Vincent, they could just get a Novavax.
Well, there is an option, but there's just no evidence that these issues are real for mRNA vaccines. The integration is just a theory. There are no data, and we've given millions and millions of people mRNA vaccines with none of these untoward effects. So,
yeah, I mean billions, right? We have great safety data here.
But I think that Laura's point is very good. Is that they don't come off as being misinformation because they reference enough places, but the way they create their narrative, you end up being suspicious. And I'm sure there are people doing this who know what they're doing, right? So
unfortunately, it's $1 billion industry misinforming people and making it seem innocent and reasonable. This is not accidental. I'm sure that document took lots of time and they spent a lot of money making sure that the misinformation was effectively conveyed.
Well, and Daniel, the bottom line here is the state says you don't have to get these vaccines, and the voters in that state love it, so they're going to vote for the governor, right? That's the bottom line. Politics.
When you mix science with politics, Vincent.
That's TWiV Weekly Clinical Update with Dr. Daniel Griffin. Thank you, Daniel.
Oh, thank you. And everyone, be safe.